Hello, I'm Arthur Brandwood and welcome to this webinar on biocompatibility and risk management and ISO 10993. Let's just begin with ISO 10993. This is a large body of standards. It provides a high-level guidance on how to conduct biological evaluation, as well as other parts of the standards with detailed test methods for investigation of different aspects of the safety and supporting guidances, and many references to other methods and guidances in national pharmacopoeia and national standards. I'd just like to point out that this has taken almost 25 years to develop this body of standardization. If we look at the set of standards that make up the ISO 10993 series, Part 1, the parent standard, of course, sets out the overall framework, and that's what I'll be talking about today, is conduct of evaluation and testing within a risk management process. But supported by that, there are a whole bunch of standards which provide test methods. There are two standards, Parts 8 and 12, which provide guidance on use of controls and reference materials and preparing samples for testing. There are a set of specialist standards on degradation products and how to identify and quantify degradable degradation products, whether the material is degradable intentionally or otherwise. There are two standards on materials characterization, Part 18 on chemical characterization, and Part 19 on physical and physicochemical morphological characterization. Part 17 provides a framework for establishment of allowable limits for leachables. Once you've conducted chemical characterization, what are the allowable limits? And then there are two specialist standards, Part 7 on the special case of ethylene oxide residuals and Part 2 on animal welfare. And then finally there is a technical report, 15499, which provides a guidance on how to conduct a biological evaluation within a risk management process and using this body of standards. This guidance, by the way, is now being written into the new version, currently in draft. It will become an annex of the new Part 1, which is currently being worked on by ISO Technical Committee 194. So here's what most people are familiar with when they think about ISO 10993. The Table A1 in the parent standard of Part 1, which provides a guidance on what aspects of biocompatibility should be considered depending on the nature of your device and its invasiveness. So along the left hand side here we have the type of device, surface, external communicating implant, what the nature of tissue contact is, and then how long the contact is, whether it's limited, less than 24 hours, prolonged, more than 24 hours but less than 30 days, or permanent. Anything over 30 days is considered permanent. And then depending on those factors you decide what you really need to consider in your evaluation. Notice I said consider. It's always been that way, but for far too long this table has simply been considered a checklist. Manufacturers have looked at the table, decided where they are, and then gone off and commissioned a laboratory to provide test reports wherever there's an X in the box. Now, it's not a checklist. The ISO standard Part 1 standard actually says testing shall not be carried out where you've got results from previous studies or there are existing preclinical or clinical data, including a history of safe use, which meet the requirements of biological evaluation. That's clause 6.2.1, and it's basically saying testing is not something that needs to be done every time. You need to look at the whole body of information. And what I want to look at today is how we do that and how we decide when we need to test or when we can use other information. And basically that is best done within an ISO 14971 framework. So here is the flowchart taken from ISO 14971, the risk management standard. It sets out the paradigm for risk management through the whole process of risk analysis, risk evaluation, risk control, and then learning from post-production information. The question is, how do we apply this to biological risk management? And what I'm going to do in the rest of this webinar is to set out how ISO 10993 and the processes contained in that body of standards map to this risk management framework. So let's start off with risk analysis. Risk analysis begins with thinking about the intended use, the intended purpose of the device, and how it's used. So it's really about understanding the device. So we need to first of all think about duration and location of contact. Consult the table A1, which tells you what sort of toxicology endpoints you need to be thinking about, but also consult regulatory guidances and product standards, which may give more specific information. And think about special factors. For example, is your product exposed to the dental mucosa, in which case you might choose to use the specific dental mucosal test methods rather than the more general mucosal test methods. And also think about potential misuse where it may affect biological safety. All of this allows you to define acceptability criteria for your risk management. Then characterize the device, characterize the materials, what's in the bulk, what additives, process aids, contaminants, and degradation products are used. Think about manufacturer data, and when you're looking at the materials, ask yourself the question, how much is present? Because one of the things that we need to determine is the dose of leachable products that are available from the medical device. That may be done through your own characterization or from manufacturer data, material supplier data, which can provide you formulations and chemistry. Do a chemical and physical analysis where required, using parts 18 and 19, the part 9 or parts 13 to 17 for guidance on degradation products, and think about compliance with specific material standards where they exist. For example, there are a whole range of material standards covering things like alloys. Then identify the hazards. So we need to say, okay, we know what leachables are available, we've done our materials characterization, we know what things can come out and may impact the tissues. So what's the known toxicity of each of those material components? What kind of toxic effects are known, and are they relevant? Of course, when you go to the literature of toxicity, you will generally find a very large literature, for example, on respiratory toxicity. And that's because the toxicology literature has a great deal of research describing respiratory toxicity in production environments. A lot of work on the effect on process workers of inhaling dusts or vapors from production environments. That's not necessarily relevant to the use of a medical device which may be implanted or placed on the skin. So think about the literature. Is the literature that you're looking at relevant to your particular case? And ask, what's the dose-response relationship? What's the availability of leachables? What's the rate and pattern of their release? And what's the total patient exposure? Then we can, having done the hazard identification, we can estimate risks. We can ask ourselves, what do we already know about the materials, about data on earlier generation devices, other similar uses of those materials? What have we got in existing in-house testing data, maybe on earlier designs? What's the materials manufacturer data? What does the literature show us? Bring all that together and understand the toxicology, look at the literature, think about use of things like toxicology databases. What are the routes of administration? What are the no observable effect levels or the lowest observable effect levels, the thresholds within the literature that define safety, and compare these to the known quantities and release profiles of your device, and use the methods in part 17 to add appropriate safety factors to determine whether what's coming out of your device is tolerable. Are the intakes tolerable? Part 17 defines tolerable intakes. We can then say, well, do we know enough? Have we established through the chemical characterization from the literature, from a part 17 analysis, that the leachables coming out of the medical device are within tolerable limits? If we can't establish that, we may then need to do testing to fill the gaps in our knowledge. We then do testing according to appropriate parts of ISO 10993 and compare the test results to the acceptability criteria that we've developed in terms of what endpoints are important, and whether the results show acceptable biocompatibility through things like cytotoxicity, irritation analysis, systemic toxicity, or whatever else is required. When we conduct the testing, I point out you need to follow the protocols and the controls as per the applicable ISO 10993 parts. Do consider product-specific safety standards and regulatory guidance as well, because that may modify what's required. And always, of course, do the final testing under good laboratory practice. You may do exploratory testing without GLP, but you need a set of final testing done to GLP, because regulatory authorities will expect that and are unlikely to accept testing that's not done under GLP. So we then ask, are risks acceptable? So we've done the risk evaluation, we've done the analysis from the literature, we've done the chemistry, we've done the supplementary testing, and we ask, have we met our acceptance criteria in terms of safety for the toxicological endpoints of interest? If we have, then biological safety is established. If we have not, then we may need to do further risk controls. So if we find that excessive quantities of a leachable are coming out a bit above the tolerable intake, or we have test data that show safety concerns, then we may need to introduce some risk controls. And we do this, we ask this question for each separate aspect of biological safety under consideration. In doing risk control, we think about the options and options analysis, and we can do a number of things. We can change a design, maybe reconfiguring a packaging to reduce levels of sterilants. We can change the materials to use less toxic materials. We can maybe introduce manufacturing controls, for example, batch releases and pyrogen testing. Or we can provide warnings, for example, labeling about known adverse responses. A very good example of that would be labeling on products which contain latexes about the hazards of contact dermatitis or anaphylaxis. And then the final step, of course, in risk management is that it's a living process, and you learn from post-production information, from placing the device in the field, in the clinic, and we learn. Monitor the device in clinical use, see what trend analyses we have. Are we seeing adverse events relating to biological safety? If necessary, we may need to update our risk assessments or change the device. And in doing so, repeat the biological safety evaluation. Of course, we may restrict that evaluation in scope to consider the changes made only, and not it may be possible to do a more limited evaluation speaking to the specific change. Now, this biological evaluation process is defined in this flowchart from part 1. It's a fairly complex looking flowchart, so here's a simplified version of that flowchart. It begins with considering the clinical application of the device and the associated biological risks as set out in the table A1. We conduct materials characterization, understand the device, and then we consider what the existing data are. What do we have from the analysis we've done in materials characterization or existing information about the device and its materials, and what do we have from the literature? And then we ask the question, have we got existing knowledge sufficient to evaluate? If we have, we can then move straight to evaluation of those risks. If we have not, we may need to do supplementary testing to fill the gaps in the knowledge. Now notice how the testing comes towards the end of the process, not at the beginning. We start by doing materials chemistry, and then we finish by doing additional biology if we need to. We then apply any necessary risk controls through changes in design or process. We may need to do supplementary testing if those risk controls introduce new unknowns, or particularly about addition of new materials. And then finally we proceed to document this evaluation in a biological evaluation report. So in summary, this risk management approach to biological evaluation is essentially efficient, it's comprehensive, and it's robust. It begins with a risk analysis of understanding the materials and the interactions, drawing on the published literature, material supplier data, comparative device experience, and deciding what you don't know. What testing do you need to add? You do the testing to fill in any gaps in the knowledge, and you can justify not conducting testing where there is sufficient relevant pre-existing knowledge. Evaluate the whole thing, that's the risk evaluation step, including any test results, quantify the risks, introduce any risk controls you need to do, and then report. Now, I'd point out that this risk management approach is well understood by regulators. This guidance from the United States FDA released earlier this year certainly completely embraces the risk management model. But the FDA in the US is not the only agency. Other agencies around the world do this as well. China particularly, I was recently lucky to be part of a delegation which did a special symposium with the China FDA, working specifically with the reviewers on how to use the risk management model, and the China FDA certainly accepts risk-based submissions and does not require complete testing, which is a complete change from the practices of a few years ago. And the same is true in Europe, and here in Australia our own Therapeutic Goods Administration very much is prepared to accept risk-based submissions dealing with biological evaluation. So let's just finish by looking back at the body of standards again, and I just want to point out that there's a great deal of change taking place. All of the ones highlighted in red here either have been recently revised or are in the process of revision, including Part 1, which is currently under a very active revision, again to extend to more modern testing technologies and to emphasize the risk management approach of biological evaluation. What's actually changing? Part 1 will incorporate that risk management annex and continue to emphasize the role of risk management in evaluation. Part 17 is undergoing a major revision where the model for determining allowable limits of leachables will be refined, and doing things like adding extra guidance on complex mixtures. Part 18 is undergoing a very substantial revision and asking the question really, where can chemistry substitute for biology? Can we do enough with chemistry to really understand the materials without needing to do biological tests? And parts 5, 10 and others are introducing new alternative testing models, including some new in vitro alternatives to animal models, and there are substantial technical updates going on. So keep a watch on what's going, get engaged with your National Standards Committee and understand the development of the ISO 10993 body of work. So thank you for joining us. This is a recorded webinar, but we'd be very pleased to have any questions on the contents of this webinar. Feel free to email your questions to this email address on the screen, help@brandwoodbiomedical.com. And if you'd like to learn more, please do follow us, visit our website, follow me personally on Twitter or on LinkedIn, or visit Brandwood Biomedical's company page on LinkedIn, or go to our YouTube channel, which you will find this webinar and a whole range of other webinars on different aspects of regulatory affairs. Thank you for joining us, and look forward to speaking to you again on another Brandwood Biomedical webinar.